speaker is Dr. Suzanne Conzen from UT Southwestern Medical Center in Dallas. Her talk is entitled GR, Understanding GR Activity in Breast Cancer.
Thanks very much, Carol. I'd like to thank the organizers and Dr. Lang for inviting me today to talk about glucocorticoid receptor, which is your body's stress hormone receptor in breast cancer. Here are my disclosures. Glucocorticoid receptor is a cortisol-activated transcription factor that, much like what Christy and Wayne were talking about, is a transcription factor that responds to nuclear hormone receptors. When cortisol is produced, or patients are given dexamethasone or prednisone in high doses, the glucocorticoid receptor is activated, it dimerizes and binds to DNA directly, as shown over here in figure 1, binding to glucocorticoid receptor response elements. But it's important to note that glucocorticoid receptor activation also works through other transcription factors. And so glucocorticoid receptor can act through other transcription factors, including the estrogen receptor, which it appears to inhibit AP1 and Jun. These interactions with other transcription factors can either initiate transcription or they can bind to repressive areas which inhibit transcription. Finally, there can be composite glucocorticoid receptor binding through both glucocorticoid receptor response elements and the partner transcription factor element. Together, the result is cell type specific glucocorticoid receptor activities and phenotypes. The other important thing to know about glucocorticoid receptor activity is that it is highly dependent on phosphorylation. The domain of the glucocorticoid receptor, called the AF1 domain, is highly phosphorylated by a number of transcription factor kinases, including the MAP kinases, CDKs, and also PI3 kinase pathway kinases. So it's important to remember that both ligand activation and phosphorylation activate this protein as a transcription factor in breast cancer. The key points I want to make today is that while you may not have heard much about it, glucocorticoid receptor expression is actually found in a subset of both estrogen receptor positive and estrogen receptor negative breast cancers. But what's interesting about it is that the downstream functional GR activities, which are mostly transcriptional changes, are different depending on estrogen receptor. What I mean by this is that in estrogen receptor negative breast cancers, including triple negative breast cancer, glucocorticoid receptor activity is associated with gene expression profiles that enhance tumor cell survival, EMT, and pro-oncogenic pathways. While in contrast, in estrogen receptor positive breast cancers, glucocorticoid receptor activity actually inhibits ER-mediated proliferative gene expression. And this has been shown by Gordon Hager, the Rosenfeld lab, and my lab, and it results in decreased breast cancer proliferation and better patient outcomes. In model systems, we have found that glucocorticoid receptor can be selectively modulated in both tumor subtypes. Much like tamoxifen, you can make mixed agonists and antagonists that are tolerable in humans and are being tested extensively and inhibit some of the activities of glucocorticoid receptor in a cell type dependent manner. Many years ago in my laboratory, we were interested in what factors that were present in serum were activating or repressing apoptosis. And in a model that we made, which is an MCF10A MYC overexpressing cell line, so it's partially transformed, we began to investigate a number of growth factors, and I show you some here. We found that insulin and EGF increased proliferation, but they didn't really have a large effect on cell survival. In contrast, glucocorticoid, much to our surprise, when given in physiological amounts, either with hydrocortisone or in pharmacologic amounts with dexamethasone, completely abrogated apoptosis in response to these stressors. What did this mean? It meant that we really pivoted from studying general growth factors to glucocorticoid receptor because the effect was so strong. What we hypothesized as a central hypothesis in the lab was that glucocorticoid receptor activity in a primary tumor and perhaps in micrometastases over time would lead to clinical relapse, whether there was like local microenvironmental stressors, chemo, radiation, or hypoxia. This activity of transcription would inhibit cell death. In contrast, if glucocorticoid receptor activity was low in the tumor cells, we hypothesized that the patient would have a clinical response which would be of long duration in TNBC, and they would have enhanced tumor cell death. One of the pieces of data that was supporting this was that there is a poor relapse-free survival seen for early-stage TNBC patients when you look at GR high versus GR low cohorts. So these are all patients who were early stage. They receive very similar treatments. And you can see that in the primary tumors that are GR low, the patients have a pretty good outcome with a median survival of about 70%. However, if the patients have GR high, and this is the highest versus the lowest quartile, you can see that they have a much poorer prognosis. And we wondered exactly whether this was because of transcriptional output or simply was measuring the amount of GR. One of the important observations was made by the Ellis lab in the UK that it was the immunohistochemical GR tumor cell assessment that actually did correlate with this poor outcome in GR-positive tumors. It was not simply that we were capturing lymphocytes or fibroblasts. So a take-home message from the first part of my talk is what factors give rise to pro-oncogenic GR activity in TNBC? It's not only the expression of the protein, it's also the ligand, and it's also upstream kinases that phosphorylate GR and make it a different transcription factor depending on which particular residues are phosphorylated. In my lab, Diana West published in 2018, looking at hundreds of human tumors as well as tumor cell lines, that glucocorticoid receptor high expression, or in the cell lines, initiation of glucocorticoid receptor activity, was associated with a number of DEX-activated pathways, including metastasis, cell survival, and invasion of tumor cells, and that these expression pathways, which are positive here, could be abrogated by mifepristone, which is a joint PR and GR antagonist, or new compounds which antagonize GR. In addition, there are pathways that are suppressed, like contact growth inhibition and cell death, which are suppressed with DEX alone, but can be antagonized in their suppression with the addition of an antagonist. Diana wanted to know what gene signatures were available for glucocorticoid receptors. She went back to the drawing board with Balázs Geoffrey from Hungary, and they looked at hundreds of patients. They developed a GR signature, which is either low or high, and divided the patients and found that low GR signature patients did much better. And this was a better separation than just using GR alone. And they then applied this to a new validation cohort. Diana then worked with Matthew Getz and Judy Bowie to show that when she applied this GR signature to patients at the Mayo Clinic who had had neoadjuvant therapy and then had either had residual cancer burden or a path CR, the GR signature was higher in those with residual cancer burden, suggesting that GR signature was important for TNBC response. In addition, there's been interest most recently in whether GR actually increases the propensity of metastasis. And in this experiment by Obradovich in Nature, MDA-MB231 cells were shown, if activated by GR ligands, to cause increased proliferation and metastases in the lung. And a very nice new paper in Oncogene by Zhang et al. Showed that in a syngeneic model of a non-estrogen receptive 4T1 cell, there was also increased metastasis of to the lung. More recently, Carol Lang showed that TGF-beta1 activates MAP kinase signaling in triple-negative breast cancers, causing specific phosphorylation of serine-134. And she made CRISPR knockout of the serine and mutated it to an alanine. This demonstrated that the mutation did not show trans-well migratory activity in response to MAP kinase signaling, while the wild-type protein of GR did. Similarly, the scratch wound assay showed a wild-type increase with the TNF MAP kinase signaling, and there was no change with the mutant serine 134A, suggesting that kinase activity is important in GR activity as well. In a really interesting new paper from the University of Utah, Conway et al. Showed with ATAC-seq taking primary tumors from TNBC basal-like and mesenchymal patients that there is a very different ATAC-seq revealing chromatin, chromatin exposure between luminal, shown in the purple and blue, and the mesenchymal basal triple negatives. And when they examined these exposed regions of chromatin, they found that the top 3 motif enrichments were Jun AP1, which is a well-known tether for GR, STAT3, and GR itself, because these tumors did not express PR or AR. And if you're interested in whether stress and glucocorticoids may induce specifically triple negative or ER negative tumors, there's a brand new paper out of 62,512 women who were followed for breast cancer incidence. And what they found is that patients who had been exposed to glucocorticoids versus those who had not had a higher risk of ER negative but not ER positive invasive cancer. So in summary, increased oncogenic GR target gene enrichment, chromatin availability as shown by ATAC-seq in primary human tumors are associated with glucocorticoid receptor signaling in triple-negative breast cancer. Therefore, there is interest in targeting the glucocorticoid receptor in, as an effective target to increase sensitivity to chemotherapy in TNBC. There's a randomized placebo-controlled double-blind phase 2 study ongoing with nab-paclitaxel and plus or minus mifepristone that's being run by Rita Nanda. And we also think GR may be an effective target in triple-negative breast cancer that expresses AR and is subjected to enzalutamide. This trial is being run by the TBCRC by Drs. Traina and Nanda. In addition, I'd like to just mention what I said in the beginning, which is that glucocorticoid receptor high-expressing tumors have a much better outcome than glucocorticoid receptor low-expressing tumors in ER-positive breast cancer. So it's the opposite result. And that is because glucocorticoid receptor and estrogen receptor interact at the level of chromatin. And when you activate glucocorticoid receptor with dexamethasone, you lose a number of really important pro-proliferative binding sites on chromatin for the estrogen receptor. And this is shown here in a paper from my lab by Eva Tonsing-Carter, where the cyclin D1, arguably one of the most important pro-proliferative genes that ER affects, gene enhancer, is actually occupied in the presence of GR and ER stimulation, such that when you look at ER CHIP, the occupancy by ER is gone when GR is activated. So GR is pushing ER off of chromatin, published in 2019 by our lab. And just one last plug for GR. Those of you who will be here on Friday morning, we believe that GR is important in lobular breast cancer as well, but the story is slightly different. In lobular breast cancer, you'll hear from poster 646 on Friday morning by our postdoc Candice Frerick that modulation of glucocorticoid receptor activity has different gene expression profiles compared to infiltrating ductal carcinoma that is non-lobular. With that, I'd like to thank my collaborators at the University of Chicago, where I spent 20 wonderful years until a couple of years ago. Rita Nanda, Eva Tonsing, Jeff Green, Jeannie Fleming, Fumi Alwapati, and my new collaborators at UT Southwestern and the patients in both locations. And at UT Southwestern, I'm fortunate enough to work with Ariella Honker and Carlos Artiega, and of course, all my funding. Thank you so much, and if you have any questions, I hope we have a couple of seconds. Bye.
Thank you so much, Suzanne, for a beautiful talk. We indeed have a few minutes. I wanted to ask you if you've looked for your GR gene signatures in, for example, cancers that arise in obese patients, because we see this pro-inflammatory cytokine activation of GR through the serine residue. And I was wondering if you've looked into that at all in your work.
So we haven't looked into it specifically, but we have a paper in Cancer Prevention Research where we looked at the effect of fat after social isolation in the mammary gland on tumor growth and TNBC. And we did see an effect that fat was pro-oncogenic in the mice that had been subjected to chronic social isolation versus group housing. We believe that it is pro-inflammatory. And I think there's a lot of epidemiological evidence that supports that.
But that would certainly be consistent with the P38 pathway that we've identified. There's a question from an audience member. Does GR repress ER expression and perhaps lead to ER negative tumors through that? Have you looked at ER expression changes when you modulate GR?
So we, at least in our model systems, have not seen a direct repression of the estrogen receptor. It hasn't come up as one of our target genes. That being said, the other way around is interesting because it looks like estrogen receptor can induce glucocorticoid receptor expression in some settings, and actually there's higher glucocorticoid receptor expression in general in ER-positive human breast cancer.
Great. Does GR associate with different subtypes of triple-negative breast cancer within that group?
Yes, Jennifer Pietenpaul's original work at Vanderbilt showed that the highest level of GR is in the mesenchymal subtype, but the recent ATAC-seq data from the University of Utah did show GR activity also in the basal subtype, but the highest absolute amount of GR is seen in mesenchymal.
Oh, that's interesting. So final question from Wayne Tilley. Although we can't see your slides from where we are, it looks a lot like the GR mechanism and the AR mechanism of inhibition of ER are very similar. So do you think that GR and AR are both sort of functioning as these sort of gatekeepers that constrain oncogenic ER in ER-positive breast cancer? Have you seen any differences between those kind of mechanisms of action from what you know about the AR work that Wayne is doing?
You know, I think we need to compare notes. I don't think there's a big conceptual difference in ER-positive breast cancer. We have shown that GR does displace ER from the cyclin D1 enhancer that Miles Brown and Jason Carroll described years ago, and it's a very, very strong phenotype. What's really interesting to me is what's AR doing in the absence of ER in triple-negative breast cancer? Is it similar to what GR is doing or different? I think there it's different.
Right, and you should ask Jen Ricker about that. I think she's worked on that quite a bit.
We
are miraculously on time. It's 3 o'clock, at least where I live. It's 3 o'clock. I want to thank everyone who gave a great talk, and despite some technical issues, we had a great session. So I want to thank all of our 3 speakers, our co-moderator, Christy, and all of our audience members. Please have a great week. Thank you so much.